If you would take a scan of your heart, it would look something like this. To now understand what this image tells us about the heart, we need to analyze the image. For this, segmentation is a great tool. These deep learning models can learn to segment shapes, like here. Luckily for the patient, this heart beats, and as you can see, its shape changes across the different images. But multiple images make our analysis more complex. And this is a key problem in medical image analysis, because with its current state-of-the-art that is segmentation, we can only globally compare the segmented shapes. So for example, we can compare their total sizes and the overall positions. But we need a more detailed shape analysis that can capture the local differences between the shapes and can measure how and where exactly the shapes vary. But this shape analysis relies on local comparisons, which segmentation alone does not offer. So, to reason at this level of detail, we need something more. We need point-wise correspondences between the images. Once we have such corresponding points, shape analysis becomes possible. And we have addressed this need by designing an approach that estimates correspondences for a few anatomically relevant points. And these points are called landmarks. And here you see an example of such corresponding landmarks. How do we do this? Well, we reason that segmenting an image and locating specific points on it are actually closely related problems. And therefore, we argue that the segmentation model has already learned relevant information for this problem as well. So by simply exploiting that trained segmentation model, our approach requires no additional training time, making it very efficient. In the next few minutes, I'll give more details of our approach, and then we'll see some exciting initial results on 2 real-world applications. So our approach consists of a few simple steps. First, we require that the relevant landmarks are labeled on one single image, so we minimize the label effort. Then, for each image, we extract its deep feature maps from the trained segmentation model, since these maps contain useful representations. Then, for each landmark, we calculate the feature similarity between that of the feature of the landmark and of the feature map of the image where we want to estimate the correspondence on. And here you see an example of such a similarity map. Finally, we locate the most similar points as a corresponding landmark. So, this is our approach in a nutshell. Let's now see it in action. And here we come back to the heart. And in this dataset, we study cardiac MRI, which is a scan taken over multiple time points during the cardiac cycle, so during the beating of the heart. At the first time point, we have 7 landmarks labeled for each patient, as you can see here. And these landmarks all lie in the same 2D plane. And we then track each landmark to the next time point, and to the next, so on all the way to the cardiac cycle. When we compare our approach to a state-of-the-art deep learning model that is specifically trained for this task of correspondences, our approach is able to achieve similar accuracy. Importantly, it does this more efficiently, since our approach requires no additional training time. And our results look like this. As you can see, our estimated landmarks are able to capture that cardiac motion, that beating of the heart. And now we move away from the heart to the lungs. And here we study 3D CT scans, where each patient has 2 scans, one at full inhale and one at full exhale. At the first scan, so the inhale scan, we have 300 landmarks labeled by an expert for each patient. And we then estimate their correspondences at the exhale scan, at the second scan. And even for this highly complex 3D problem, our approach is able to achieve state-of-the-art accuracy, but again, more efficiently. And these are the results for 3 landmarks, where we have the given landmarks and our estimated landmarks. And as you can see, it's remarkable that across this entire 3D scan, our estimations are so precise. And with these initial results, it's clear that our approach has potential. Its key advantage is its efficiency, since it uses a trained segmentation model. This is still work in progress. For example, we're investigating how we can manipulate the segmentation model to improve our results further. And with our approach, we can move medical image analysis beyond segmentation to landmark-based shape analysis, allowing us to understand even beating hearts.